Okay, good morning, everyone. Today we're going to be talking about histology, the study of tissues. It's actually a short lab. It's short in the normal semester as well as today. So, and the reason is in the summer we need to combine a couple of labs, and so normally we do this when we are learning about microscopes. So it's just a good opportunity to practice that skill of focusing. But today in the normal semester, it stands alone. And when it's combined with a holiday week, a lot of times we will have guest students joining us in real lab. So it helps to have a shorter lab. So you'll get out of class early today and be able to start that weekend a little earlier. Okay, so there are 4 main tissue types. There are epithelial tissues, there are connective tissues, muscle tissue, and nervous tissue. We're going to save muscle and nerve tissue for when we talk about muscle and nerve in separate labs. So today we'll just be talking about epithelial tissue and connective tissue. We're going to start with talking about epithelial tissues separately. And then I have a combined PowerPoint that will review epithelial tissues again, and then kind of compare and contrast them as we introduce connective tissues. So go to this one. So this is an introduction to histology. And so histology is the study of tissues. And as I mentioned, there are 4 major categories: epithelial tissues, connective tissues, muscle, and nervous tissue. With epithelial tissues, they are going to be in layers. And so those layers are arranged so that the cells are on a basement membrane. And they are named by whether or not those tissues are in one layer or more than one layer in general. We'll talk about pseudostratified by itself. If it's in one layer, its function is usually absorption or diffusion. So we'll see our first tissue is going to be simple squamous epithelium, and it is, sorry, I'm going to have to, and it is going to function in the diffusion of gases. So it'll allow oxygen and carbon dioxide to be quickly equilibrated since it's only one layer thick. If those tissues are arranged in more than one layer, then the function is usually something along the lines of protection. So we'll see that, and our sample comes from the esophagus, where you have to have some protection of underlying tissues because you have food going down that passageway. It's also in like the skin, where you want to form a protective barrier from the outside environment. Pseudostratified has only one layer. All cells do touch the basement membrane, but it appears to have more than one layer because, as you'll see, the cells get a little bit more jumbly. They don't align nice in a nice straight line. Okay. So the way we name a tissue, we start with the number of layers. We say simple something or stratified something. And so then the second term comes from the shape of the cell. If it is kind of this irregularly flattened shape cell, we call it squamous. So as I mentioned, our first sample or our first specimen is simple squamous epithelial tissue. Then our second one is going to be simple cuboidal epithelial tissue. And this is one layer of cube-shaped cells. And cuboidal cells are going to be shaped like a cube. And the other thing to look at is what shape is the nucleus. So in cuboidal cells, the nucleus stays nice and round, and it stays centrally placed in that cell. In a squamous cell, the nucleus starts to get flattened out, especially if there's more than one layer. We'll be also looking at a simple columnar epithelium in the small intestine and the jejunum, and these are going to be rectangular cells. And then we'll look at a transitional epithelium as well, where the tissue changes shape. Okay. So this is just to review that you could have simple squamous, a single layer of squamous shaped cells, or you could have stratified squamous epithelium, where you have multiple layers of squamous shaped cells. As I mentioned earlier, epithelial tissues are adhered to this basement membrane, to a basal surface. So they have polarity. They have a top and a bottom. So this is called the apical surface. It could be facing like the exterior, like the outside environment, or it could be facing a lumen. So you could have, we'll see like a kidney tubule that contains urine filtrate. This could be the lumen of your GI tract. And then the basal surface is that bottom surface. So each cell has a basal surface and an apical surface. This just is a kind of a review of what the nuclei are going to look like. This gives you kind of a hint at maybe what type of a cell you're looking at. Cuboidal cells are very square. Their nucleus stays nice and round. And columnar cells are these rectangular shapes. Their nuclei stay pretty round. And a simple columnar tissue will have them all lined up kind of in a nice little row. In pseudostratified tissue, that's where it changes. The nuclei start to become this wavy line. These are the epithelial tissues that we're going to go over. And in this slide presentation, it's more of a, here are some textbook examples. And then in the next PowerPoint are the actual slides you would be seeing in lab. So I've taken those slide specimens and instead transferred them to a PowerPoint. So this is just, these examples are not, they're just from a textbook. This is just a presentation. It's not what you would be seeing in lab. It's just to give an extra review of epithelial tissues. I found in the past that just a quick overview of epithelial, the next PowerPoint is not enough for most students to get comfortable with epithelial tissues. So I added this one in because a lot of it, mainly because we have time to do it. So you kind of get epithelial tissues twice, just they're a little more complicated. We will look at 6 of these. We won't be looking at stratified cuboidal. Okay, so that one gets off the list. But we'll be looking at the other 6. And as I mentioned, we start with simple tissues. We'll start with a single layer of squamous cells that are found in the lungs. Then we'll go to a single layer of cuboidal cells found in the kidney, and then a single layer of columnar, which forms epithelial tissue found in the small intestine. When you're looking at simple squamous epithelium. This is both the basement membrane and the cells are on top of it. We would need to get a higher magnification to be able to see each of the individual cells. But it's like this is a nucleus, and this is a nucleus, and this is a nucleus. So this is a structure that's made up of 20 to 30 cells that are all on a continuous basement membrane. Okay. These are macrophages, so you can just ignore those. So white blood cells that are in the alveolar sacs. So when we look at simple squamous, it's these kind of like pink squiggly lines with little dots that represent the nuclei of individual cells. For both the quiz, which is next Thursday, and the lab exam, which is in 2 weeks, you need to be able to, so you'll be given a slide like this, and you'll need to identify, is it an epithelial tissue? Is it a connective tissue? Which epithelial or connective tissue is this? So this is simple squamous epithelium. And then where would you find it? Okay, so this one is found from the lungs. You do not need to know specific functions. You need to know general functions. So you need to know that like something that would be a simple epithelium would be a great tissue for absorption diffusion, that a stratified epithelium could be used in protection. So general categories are helpful. When we get to connective tissues, we'll talk about like the function of collagen fibers and elastic fibers, but nothing that is terribly specific. So just general categories of function. The other thing you'll need to know is vascularity, but that's something that we're going to go over on the next presentation in greater detail because it varies more with connective tissues. So it's kind of the fourth big component. This is the classic representation for simple cuboidal epithelium. It is where this thin purple line here is the basement membrane, and then depending on where you catch that tubule, because these are kidney tubules, you may or may not be cross-sectioning the nuclei at the same time. So even though you have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, like 11 or 12 cells around, you're only catching 4 nuclei. But there are you can kind of barely define where these cell definitions are. They end up kind of looking like squashed donuts. But compared to other tissues, this tissue type does have a very distinguishing look. There aren't that many places you find simple cuboidal tissue in the body. So kidney tubules are often used as an example. In 1248, you'll learn to distinguish the different types of tubules. So we start you off with just identifying a single simple cuboidal epithelium in a kidney tubule, and then next semester we'll take you one step further to distinguish the difference between different types of tubules. This is an extreme magnification of simple columnar. The sample you would be seeing in lab on the next presentation is not quite at this magnification. So this is just to illustrate how so you can see a more defined boundary between cells, like you couldn't see in the previous slide. So this is simple columnar epithelium. They are attached at the basement membrane, and then these nuclei, they stay spherical and they line up along that basement membrane. And this one illustrates how, slides are going to be presented in different orientations. Remember how that E turned upside down? So it turned into an upside down E. Well, sometimes your basement membrane may be at the top of your screen, and then that apical, or the top of your cell, is actually at the bottom of your screen. So it's that way with cells too, because if they're lining a lumen, they're not always going to be, where this is the bottom and this is the top. So when you're looking at a slide specimen, find where the basement membrane is. It's usually where that nucleus is, and then the apical, that top surface, is going to usually be the more rounded side. So simple columnar epithelium, this one is found in, so our samples, our specimens, if you're asked for a location, our location is the jejunum, it's the small intestine. Our fourth epithelial specimen is still a simple tissue. It's still only one layer, because these cells, even though they look a mess, they all still attach to this basement membrane. So it's called pseudostratified, a false sense of stratification. The telltale sign of pseudostratified, or are pseudostratified, is that it is ciliated. So we have a ciliated pseudostratified columnar epithelium. And so the cells will have a basal attachment to that basement membrane. On their apical surface, they'll have these cilia. And in both simple columnar epithelium and in pseudostratified ciliated columnar epithelium, you will find goblet cells. Goblet cells secrete mucus. So in the preceding tissue, the mucus was used for protection. In this tissue, that mucus is going to serve to entrap particles. So you may be entrapping pollen or air pollution, or smokers, they'll get smoke particles entrapped here in the mucus. And then the cilia will always beat in an upward direction, and that carries the entrapped particulate, and then it dumps it into the esophagus, which then it gets carried down to the stomach and digested. In chronic smokers, these get burned off, they get singed and burned, and so there's no cilia available to beat that mucus up towards the esophagus, and that's where that smoker's cough comes from, because the mucus settles down into the respiratory tract. So pseudostratified ciliated columnar epithelium is found in the trachea here and on our slide specimen. Okay, so we're going to move on to a couple of stratified tissues. So stratified meaning multiple layers. The one we are looking at now and in lab is going to be what's called non-keratinized, and they are found in the esophagus. So this is the basal surface. So we've got a basement membrane, and underneath this basement membrane is connective tissue known as the dermis, and it is irregular. Okay, so because that dermis has an irregular surface, it causes a wavy basement membrane. So this underlying tissue is wavy, which creates an increase in surface area, which is helpful for the diffusion of oxygen and nutrients. Because, as you're about to find out, epithelial tissues are all avascular, meaning they do not have their own blood supply. The blood supply lies beyond the basement membrane and lies down here. So by increasing that surface area, it allows better diffusion through the multiple layers. So this basement membrane is wavy. That means that this first layer of stratified squamous epithelium is also wavy. So you get a wavy layer of stem cells, baby keratinocytes, basal cells, and when they divide, they leave one cell at the basement membrane, and then a daughter cell is going to continue into the next layer. As we get new layers, they push upward, and as they get further and further away from the blood vessels down here in the dermis and the connective tissue, they will start to die. And that's where we start to see this flattening of these nuclei, that some of them will start to disintegrate, and then this layer will start to slough off. So when you're identifying stratified squamous epithelium, you'll notice it's highly cellular, many cells. These cells start off with nice round nuclei indicating nice healthy cells. And then as the layers progress, they become fewer and farther between them, and they flatten out. So that's how you'll identify a stratified squamous epithelial tissue. Our last epithelial tissue that we're going to be looking at is transitional epithelium. And it's in the next slide presentation. I have examples of it both contracted and not contracted. So this is found in the urinary bladder, and this is in its relaxed state. You can see that the basement membrane is relatively even. And then some other defining characteristics are if you go to the most apical, the most outermost cells, these nuclei are still spherical and they're still relatively evenly spaced. You don't, start to really notice huge differing numbers. I mean, they're a little bit different, but not like in the previous slide. These are because these are still healthy living cells. And then the other defining characteristic of transitional epithelium is that they have this kind of mushroom cap, this domed cell appearance as they in this relaxed state. Okay, so dome shaped cells, spherical nuclei on the apical surface, and just kind of more evenly dispersed. So this is just a little mini quiz. I think you'll be able to go through it pretty quickly and easily, and then we'll move on to our compare and contrast. Okay, so these are all epithelial tissues, so that you kind of get that first one off, and then the tissue type, where in the body the tissue is found. Those are the next two big identifiers that you would need to know for a quiz and for your lab exam. Okay. This is not the slide that you will see on your quiz or your lab exam. Those will come from the next PowerPoint. This is one that is much more blown up or magnified, but it is to illustrate the shape of the cell and how many layers. So the name of this tissue is you have a single layer, so it is simple, and the shape squamous. That's right. So simple squamous epithelial tissue found in the lungs. Yes, very good. Okay. This is looking from the surface. So going on to this one, this is the basement membrane. We have another single layer, and these cells are in a different shape. So we now have simple cuboidal. Very good. Simple cuboidal epithelium. And these are found where? In the kidneys. And I'll just say right now that they're found in the kidney tubules, because you're going to find out that there are other cells found in the kidneys. Like I said, we're just building you up for 1248, where you're going to, histology is a large part of 1248. I will just introduce that concept to you now. So this is one of those labs that you don't just like learn and forget. Histology will stay with you for the next semester. It is built into pretty much every lab in 1248. You'll learn histology of the reproductive system, histology of the urinary system. So you'll have quite a bit more histology in your future. So this one is more almost of a graphical representation, but again, you have a single layer. This is not the greatest. This is probably my least favorite slide, but it's supposed to be a single layer of rectangular cells. So this would be, yes, simple columnar, and it was found in the it's found in the jejunum, which is part of the small intestine, yes. And on the quiz and on the lab exam, I'm not sure if it says jejunum or small intestine. So be familiar with both of those. Oh, there we go. Okay. So this one, these are the giveaway. These are our cilia and our goblet cells, which you'll see goblet cells in several other tissues. So those are just other cells, but these little cilia, we only have one type of ciliated tissue. Yep, that's right. Pseudostratified ciliated columnar. There we go, epithelium. I know, it's a very long name. Thank goodness it's multiple choice. You don't have to write it in. Okay, so, and it's found, sorry, where is this found? In the trachea, yep. Sometimes you'll see trachea, sometimes you'll see upper respiratory tract. I do believe that trachea is listed on both the quiz and the lab exam for this one. Okay, so this one, you have many layers. And you notice the arrangement of nuclei. What kind of tissue does this represent? So the names are going to always start with however many layers. Stratified. And then they get squishier as they go from younger to older. So it is a stratified squamous epithelium. So stratified squamous, they'll start with these little circular nuclei, and both the cells and the nuclei are going to flatten out as they progress towards this apical or an older surface. Where in the body would you find this tissue? For our slide samples, it's in the esophagus. Both of the slide, all the slide specimens are going to be coming from the esophagus for this particular tissue type. You'll learn about keratinized stratified squamous when you get to the integumentary system in lecture. Okay, so this one you get two because it goes between two states. Whoops. Sorry. It is transitional epithelium, and it is a stratified tissue because it has multiple layers. The next slide presentation has a better representation, and I think in the, when we introduced it has better. This is just showing it has these dome-shaped cells. Again, it's heavily magnified. But where in the body would you find this tissue? It's found in the urinary bladder. Yes. Okay. So the difference between this stratified tissue and the other one is mainly on the arrangement of nuclei at the basement membrane and at the apical surface. And on the next PowerPoint, just like 5 minutes, I have them side by side so that you'll be able to kind of illustrate the differences between the two. Okay. So I'm going to go to that one. So on this PowerPoint, since we've had an introduction to epithelial, now we look at epithelial tissue versus connective tissue. So epithelial tissues are pretty consistent. They all have this basement membrane. They are all avascular. And this is something I need to kind of have you star, point out, write 100 times. I don't know, like make a giant flashcard, because even in we'll talk about the histology lab review, they'll say there are some sources that will say that some of these tissues are vascular, and they're not. If you look at your textbook, there's been now just a real, we're trying to all get on board that epithelial tissue is avascular. Okay, it can be in structures that are vascular, structures that have a large number of blood vessels that might be close nearby. But epithelial tissues are all avascular because the tissues are on one side of the basement membrane, and then the blood supply is on the other side. So all epithelial tissues are avascular. They do not contain their own blood supply. They do not contain their own blood vessels. Okay. So make sure that, because this is a term, this concept seems to not be getting clearly expressed, even as you'll see later on. Okay, so all epithelial tissues are avascular. They do not have their own blood supply. They reside on a basement membrane that gives them this polarity, this top and this bottom surfaces. They'll often have modifications like cilia and goblet cells. They form linings, and they have lots and lots of cells. Okay. Connective tissues are the catch-all category. So if you're not an epithelial tissue and you're not muscle tissue and you're not nervous tissue, then you are a connective tissue. So connective tissues are a diverse category of tissues. Connective tissues can be fluid like blood. They can be very hard like bone. They're going to include things like cartilage. They will be kind of squishy like adipose tissue. So connective tissues are the catch-all category of everything else. So what we do is we give you an illustration of 7 of them. We could keep going, but we'll show you 7 of them to illustrate different properties. And like things like blood, you'll get, you know, later date. You'll even get more about bone at a later date. So we'll give you 7 examples to illustrate some of their different properties. But because they are varied, they have no basement membrane. They have fewer cells, but these cells are specialized. And because these are so uniquely different from each other, their cells are uniquely different from each other, and they get their own names. And you get to learn these names. So instead of, you know, so this is one of the things that you do need to focus on. I think it's one of the new focuses of this lab is to get more familiar with the types of cells involved in connective tissues. Okay. And these cells secrete fibers, which are made from proteins. And then they also secrete the matrix in which they reside and these protein fibers assemble. So we'll start with a quick review, and I will show you these are, so if you were in real lab, what we would have is we would have 13, 14 or so microscopes set up with slides on them, and you would be progressing through the microscopes looking at slide specimens. And it really kind of varies year by year which ones we select to look at. So that's why some of these have multiple images, because I never know quite which one we're going to pick from year to year. Okay, so the first slide specimen that would be on a microscope, that you would be looking through the microscope, and what you might see would be simple squamous epithelium, and it looks like a pink squiggly line. This is the slide that you'll see in 1248. So just to kind of like that, I like to start bringing in, like, why am I learning this? What am I going to need to know this for? Well, we will tie this into the respiratory system next semester. So this is what we're focusing on now, to learn that it is a simple tissue made up of one layer of squamous cells into simple squamous epithelium, but it's going to be forming these alveolar sacs that are going to be part of the respiratory system and functioning in gas exchange. So This is usually the more common one seen in the histology lab, but this is the one that is seen in 1248. So, and this in the origin, it would be lung tissue. For simple cuboidal, this is kind of like, this slide here is really a textbook one. It doesn't look like that in real life. Okay. This is the slide that you would see in real life. This is taken from, I would guess probably a monkey, maybe guinea pig, I don't know. But this is a kidney tubule here. This is a glomerulus. And so the glomerulus tells you that you're looking at a kidney, the kidney structure. And this structure here is the kidney tubule. For your quiz and lab exam, you might be seeing this one because it is a greater magnification focusing on the tissue type. But you should also be familiar with the bottom slide because that is the one you'll see with the urinary system. And it will, because these are those kidney tubules, and it's the glomerulus structure that lets you know that you are in the kidney. Okay, so you do need to spend a fair amount of time with these images, knowing which one represents each tissue type. For simple columnar, you would be given a slide specimen that looks very similar to this one. It is so that you can see these are arranged in something called a villus structure. They look like fingers. Digestive juices get kind of trapped between the fingers so that they are there for the nutrients to be absorbed on this. This is the BB stands for something called a brush border. It's an extension of their plasma membrane. But so simple columnar, you can determine it's simple columnar because these cells line up on a single basement membrane, and then the nuclei are in a straight little row. So this is simple columnar epithelium, and it's found in the jejunum. With pseudostratified ciliated columnar, yes, it's a mouthful, epithelium, the distinguishing characteristic is really going to be these cilia. So the slide specimen that you're given is probably going to look like this one, or it's going to look like this one. But there's going to be a huge focus on showing you that there are cilia, because we're not trying to trick you between these two. We're really trying to emphasize that the ciliated feature in pseudostratified tissue. Okay. And then it does have these, you know, the nuclei don't line up nicely. Okay. But pay attention and look for the cilia, and it's found in the trachea. Okay. Now this is where your question came up, how to tell the difference between two stratified tissues. You start by looking at the basement membrane. This one here is not a great one. So this is a better example of a stratified squamous epithelium. Usually the basement is significantly folded. So you've got a wavy basement membrane to start with. And then the other telltale sign is you will see these tightly clustered round nuclei at the basement membrane, because these along the basement membrane are stem cells. So these are rapidly dividing, very healthy cells. So you have these nice healthy round nuclei. This down here, this is dermis, this is connective tissue at the bottom. Okay, so where you get these dark, round nuclei. This is the basement membrane. And then you have layers above it. And in the layers above it, the nuclei have flattened out. These layers have flattened out, and everything starts looking very squished. This one, the basement membrane, is further down here. And these nuclei stay rounder. And then here it's just the nuclei get a little less dense, okay? But in general, they'll also just start getting flatter. Okay. In a transitional epithelium, this was one of the slides we used to use. We've switched more to a slide that looks like this. The cells at the apical, at the top surface, they have this round, almost fluffy surface. And then they have spherical nuclei that are interspersed throughout all of the layers. You never see dense, 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 and then sparse, sparse, sparse. It's, even in the contracted state, they're kind of evenly spaced. Usually for transitional epithelium, it'll be a slide from the relaxed state because we want to illustrate those domed cells. So those are the 6 epithelial. Now we're going to look at 7 connective tissues. As I've done in bold, you need to know what the cell type is for each of these connective tissues and then where they're found. There are a few that will say you don't really need to know this location. We start off with one of those. That would be the loose connective tissue. It's because it's an underlying tissue. It's in fascia, it's in dermis. It's kind of one of these tissues that's found in many different locations, but not really in a specific location. It is the most common connective tissue. Its cells are called fibroblasts, and they are going to secrete collagen fibers. So collagen fibers are very strong. They also tend to stain this kind of mellow pink color. We will also see some elastic fibers. Elastic fibers are going to stain a darker purple, but they still have a fairly significant weight to them. And then reticular fibers are these tiny fibers here. They'll actually usually stain a dark purple. Sometimes they'll even stain almost black. And they kind of form this like a guy wire arrangement. They're very kind of strong, but they're not very flexible. So loose connective tissue is found everywhere because it has just the right amount of structure and no structure. It's a very flexible, used tissue. It has this kind of crosshatch design where in the dermis, you feed blood vessels through it and you feed nerves through it and you can secure hair bulbs to it. So it's just this great tissue to stick things in that you want held but not like firmly secured. Okay, so it's this multi-purpose connective tissue. It would be considered to have a moderate level of vascularity because it does have those blood vessels that run through it. Dense connective tissue, its cell type are also fibroblasts, but they secrete almost exclusively collagen fibers. And these collagen fibers are pretty large in size. You can actually, if you blow it up magnification high enough, you can actually see them. And the way they're found in tendons. So this slide is found in tendon. And the way that these collagen fibers stack, they stack kind of one on top of another, and it forms a very strong structure. But because they pack so densely, they have a very low level of vascularization, which is why tendons and ligaments heal so slowly, because there's very little blood supply actually to a tendon because of this tight packing, but it gives them incredible strength, and also they're resistant to changes in pH. So they can resist high changes in temperature and high changes in pH. They're a structural element. So dense, and you'll see that there's later on you'll come across dense irregular connective tissue. This is dense regular connective tissue where they're aligned in a regular fashion, one on top of another. Okay, so we have loose areolar connective tissue with this little crosshatch design, and we have dense regular dense connective tissue with its very linear layering of collagen fibers. So we've gone from two very pervasive connective tissues and that can be used in multiple ways, in multiple locations, to highly specialized connective tissues. So adipose tissue and bone tissue are both very highly specialized. They are both storage tissues. This will be used for energy storage, and then this will be used for calcium phosphate storage. This will also be used structural, of course, in the skeletal system. But adipose tissue is going to be used for storing lipid droplets. Each one of these is a single adipocyte. A single adipocyte, a single adipose cell. And so this entire slide is adipose connective tissue. This is a blood vessel here. Okay, so this is a blood vessel, and this here is a nucleus. This is a single cell right here that belongs to that nucleus. Inside is lipid, it's oil, and it is pushing out on the cell membrane. So it gives it a very, very spherical look. All of these cells are spherical. Adipose tissue often gets confused with another tissue. Can anyone guess what tissue type, when people see this slide, sometimes they might think it is? We have seen it twice. No guesses? No, it's an epithelial tissue. Some people think this looks like simple squamous. Some people think this looks like the lung tissue. Okay, so don't get those confused. Obviously, you guys don't, so that's good. Maybe it's different when you actually look through a microscope, but when you're studying, because it might be, be a couple weeks to your lab exam, but with adipose tissue, these are spheres because they contain lipid droplets, they contain oil. So these are spherical, this is a nucleus, and then these are blood vessels. So this is a vascularized tissue. And each one of these is its own cell. For bone, the specialized tissue type are osteocytes. And each one of these dark spots is called a lacuna. It's the home for an adipocyte. So an osteocyte. Sorry. So for each one of these, you would have an osteocyte in there. So we would have osteocytes living in each of these little lacuna. So the cell type for bone are osteocytes. You don't need to know about the rest of the structures. You'll learn about that in lecture with the skeletal system. It does stain brown. It's arranged in this tree trunk type of structure. This is dense, compact bone. There is also spongy bone that we don't use in this lab, but you'll learn about in lecture. Oh, and bone is also highly vascular, by the way, because this is the central canal, or there is a central canal, and it has an artery that's going to be going through. See, version canal is kind of an older term, but central canal is a common term, and it'll have an arteriole, a venule, and a nerve going through. So you'll find the central canal, and we'll talk about that in lecture. Okay, so there has been loose and dense connective tissues, areolar, and adipose and bone. So we've got loose areolar and dense, and then we've got adipose tissue and bone. And now our last three are cartilage tissues. All types of cartilage tissues in all three of our specimens, the tissue type is going to be chondrocytes. So chondro is going to indicate cartilage. Okay, so, and chondrocytes look like fried eggs. They often will double up and pair together. Okay. So you might see a single fried egg, you might see double fried eggs, but anytime you see this type of a structure, these are chondrocytes. These are the cells of cartilage. So if you see these chondrocytes, you know you have cartilage. Now we have three types of cartilage. We have hyaline cartilage, elastic cartilage, and fibrocartilage. This is where the fiber type and how fibers stain is going to come into play. So hyaline cartilage, most common because it is slippery. It's found on the edge of bones. So if you've ever taken like a chicken leg, like a raw chicken leg, and you've like pulled it apart and it's shiny on the end, that is hyaline cartilage. So hyaline cartilage is slippery and it's shiny and it's very good at reducing friction. Our slides are from the trachea. Okay, so the tracheal cartilage rings have been sectioned to expose the hyaline cartilage. So for hyaline cartilage, you have chondrocytes, and they are secreting itty bitty, teeny tiny collagen fibers. They're so small that they you can't make them out in the matrix. And so this starts looking like pink jello. Okay, it's like a pink gel. So hyaline cartilage, you figure out that you have it by first identifying chondrocytes, so you know you have cartilage, and then it's this very mellow pink lavender jello matrix, and that will indicate that you have hyaline cartilage. Cartilage is avascular. Cartilage does not have its own blood supply. And the slide specimens, they tend to pretty much all look like this. Hyaline cartilage is nice. Collagen stains well. And it's always kind of this pretty pink color. Just to let you know, pretty much all the structures internally, most of your tissues are usually white. We aren't pink, purple, and dark purple inside. These are stained for their properties. I wish we were as interesting on the inside as histology makes us look, but we aren't. We stain for contrast. Okay. Moving on to elastic cartilage. Again, if you identify something that looks like a fried egg, you have a chondrocyte, so you have cartilage. So elastic fibers tend to stain very dark purple. It can look almost black. It's really like an intensely dark purple. I do miss a little bit that you don't get to see these on a microscope, because it's just sometimes kind of, I don't know. I really like histology. I like chemistry too, but and they tend to wrap almost in the figure 8 around their chondrocyte or a couple of chondrocytes, and they're found in the outer ear. It's because this is elastic here. Okay, and so they form these elastic structures with large chondrocytes and these dark, heavy elastic bands around them. Our last specimen of the day is fibrocartilage. So fibrocartilage, it's harder to stain fibrocartilage. So sometimes you'll find it stained blue. Sometimes you'll find it stained kind of a strange pink color. It's very good at resisting compression, so you'll find it in intervertebral discs. The chondrocytes are kind of tiny. They're a little bit harder to see. It is the only slide specimen we have that's staining blue, but normally don't try and use color too much, but in this particular lab, it's a little bit helpful. And that collagen tends to be that pinky color, and elastic is that dark, intense purple. Here we have waves of collagen, and they're staining either blue or pink, which then kind of makes them look purpley. So this one color is a little bit maybe not ideal to go upon, but they do have this, it has this wavy structure. And like I said, it resists compression. If we give this specimen to you, we do try and still show you that there are chondrocytes, just so that you have an idea that you're looking at cartilage. And again, found in intervertebral discs. Okay. So before I talk to you about like the pages in your lab manual and the histology lab review, are there any questions on these two PowerPoints? Oh, what do the numbers represent? So these numbers represent the magnification. So if you were looking, if you had that low, so when you see this number, it usually is not taking that ocular magnification into effect. Okay, so it's the same thing on the elastic. Oh, numbers on the elastic. There's a 1 and a 2. Numbers on the elastic cartilage slide. Oh, okay. So for 2, this is showing that this is an elastic fiber, and for 1, this is showing that this is a chondrocyte. Yep. So on the elastic cartilage slide, 1 is showing the chondrocytes, and 2 is showing the elastic fiber. Are there any other questions on these? So we're going to talk for just a second about, okay, so you have your lab guide over here. Okay. And so on this slide, presentation also says to like make sure you complete pages, I think 28, 29, and I'm going off of this. So it might, if you happen to have a hard copy, I think it's pages 20 and 21. So on some of the PowerPoints, if the page numbers are off, just kind of go with it, because I'm going from, some people have a white lab manual, some have an orange lab manual, and some people have it online. So it may not completely match perfectly, but you should be able to kind of figure out from that, you know, being on a topic. Okay, so again, these represent where they are found. These are going to be kind of the things you follow. And then this is the official lab activity, but you don't have to do it this way because you don't turn this into me. This is for you to study. So if you want to do this on a document, if you want to do this on flashcards, however you want to do this so that you can use it to study, whatever works for you is how you should be doing this. But for each of these, you need to pull down the images so that you can match the image to the name. You do need to know from the image, is this an epithelial or is it a connective tissue? You need to know, is it vascular or is it avascular? So if it's epithelial, it's automatically avascular. If it is a connective tissue, it could be vascular or avascular. So a little bit of detective work in that one. Where did it come from? It's on the slides. It's also in the preceding pages on the lab manual. Special characteristics, that's to help you identify that tissue. It could also be like for pseudostratified ciliated columnar, it has cilia, it has goblet cells. Transitional, it has these dome-shaped cells. For once you get to your connective tissues, you need to make sure that you are putting down what is the cell type. So for cartilage, you have chondrocytes. For areolar and dense regular, you have fibroblasts. For adipose, you have adipocytes. You need to make sure you know those cell types. So on whatever lab book, or if you want to do a, like I said, flashcards, however, Quizlet, whatever format you want to do. Just make sure you know that information, that you have it down to reference, that you are going through that. Yes. So like, yeah, depending on the lab guides, it's like I said, they're different on different pages. So just adjust accordingly. Okay. See, here we go. Make sure that you are reading. So especially for the lab exam, okay, make sure that you are reading that lab manual, because the lab exams are taken in mind that you are reading the lab manual and that you are going through these exercises. So make sure that you know, like, all epithelial vascular. So if there's ever contradiction on anything, this is the defining, go with the lab manual. These are what we are trying to, keep the concepts that we're trying to emphasize. The cell types and the fiber types. You don't have to know about fibrin. That's we're not doing that one, but it should be collagen, reticular, and elastic. Okay. Okay. So the last file that we have is okay. So there is the exam review. That's that PowerPoint file. I'm going to go to this one. So you have this one here. Okay, it's a review with this file. You can pull it up as a PowerPoint, you can pull it up as a PDF, whatever document format you prefer. They're both here. This lab test review podcast goes over it. It gets highly redundant if I go over this again with you right now. Okay, so you need to kind of go and do the exercise of putting that information together on yourself by with yourself, or if you happen to have a study buddy, that'd be great. If I go through this right now, it's super boring and it's really redundant. So so that you don't have to sit through that for the next 40 minutes. There is a podcast that goes through that for you, but it has the one thing it's not correct on is vascularization. So it's great. I'm grateful for it, except for these two corrections have to be kept in mind, that all tissues are avascular. They do not contain their own blood vessels. All blood vessels are outside the basement membrane, and connective tissues can be highly vascular, avascular, or somewhere in between. Other than that, great. Okay. Like I said, this concept has been modified a bit here and there, and this podcast was before kind of we solidified our definitions of what we meant. So do that at some point between now and next Thursday. Also complete that lab manual activities between now and next Thursday. And then I will be sending you an email about next Thursday as well, because there's a series of videos. I recommend watching them before lecture. I mean, you could do it after lecture, but I'll send you a little email about that as well. Okay, so are there any questions about what you need to do between now and next Thursday? Are there any questions over the content? If not, class is dismissed, and you can stay if you have other, like, we don't have lab the week of lab exams. That is correct. You need to take your lab exam somewhere between Monday at 8 a.m. and Friday at 10 p.m. And like I said, I do not recommend waiting until that last half hour, just in case you have technical problems. So we will not meet via Zoom the week of lab exam. So we will meet next Thursday, so in a week, and you will have a quiz on the 17th. And then that following week, we will not meet for lab lecture on by Zoom. You will just have your lab exam. There are no external assignments that you turn in. So you won't be turning anything into me. If you do have a question that comes up, just email that to me and I'll email it back to you, or we can, if it's something that is more visual, I can always set up a Zoom meet. But yeah, it's just studying on your own. Yeah, the lab exam is highly visual. Lab quizzes are too. So a lot of it is really identifying the structures or slides. But you do have to study because you won't have time to like try and figure it out. That's where students get in trouble, is if they think, oh, I'm going to have time to figure it out. That minute per question goes by really quick. Plus, we do build on this in 1248. So you do want to know it. Any other questions? If you think of anything, can bring it up next week too.
Actually, I have a quick question.
Yeah.
So